Hello, my friend. Today, we from Group 3 want to tell you about the kingdom Monera. So the first question is, what is the difference between virus and bacteria? For the viruses are not living organisms. Viruses only grow and reproduce inside of the host cell they infect. When found outside of this living cell, viruses are dormant. Their life therefore requires by hijacking of the biochemical activities of living cells. Viruses are submicroscopic. A viral infection is systematic. Virus infect a host cell and then multiply by the thousands and leaving the host cell and infecting other cell of the body. Systemic disease caused by viral infection include influenza, measles, polio, AIDS, and even COVID-19. For the bacteria are living organisms. Consist of single cell that can generate energy, move, reproduce, and make its own food. This allows bacteria to live in many places, such as soil, water, plant, and animal. Bacteria are more giant than virus. Bacterial infection is usually confined to a part of the body called localized infection. Infection may be caused by bacterial or endotoxin produced. Bacterial diseases include pneumonia, tuberculosis, tetanus, and food poisoning. That's all for me. Let's move to the next question. Bye.
Next, we go to the question number 2 is about archaea versus eubacteria. So archaea did not have peptidoglycan in cell wall, while the eubacteria have peptidoglycan in cell wall. And archaea can survive in extreme environment or harsh environment, like very salty or very hot. But the eubacteria cannot survive in that situation, but they usually easily found everywhere on the earth. And archaea interns are present, but eubacteria are absent. For example of archaea is thermoplasma and eubacteria are bacillus. So for archaea bacteria there are 3 types, which is methanogens, halophiles, and thermophiles. And the eubacteria has 2 types, which is gram positive and gram negative.
Hello. Next, I will continue with my question. My question is about one example each for the thermophiles, methanogens, and halophiles. Let me explain briefly about these 3 things. The first one is the thermophiles. A thermophile is an organism that is adapted to live at relatively high temperature between 41 degrees to 122 degrees Celsius. Thermophiles are mostly from the domain archaea. Let me show you one example. For example, the tag polymerase used in PCR. The second one is methanogens, which produce large quantities of methanol during the decomposition of organic matter. One example of methanogen is methanococcus. And the last one, the third one, is halophiles. A halophile is an organism including bacteria, archaebacteria, and some eukaryotic organisms that live in hypersaline environment with different salinities. One example of halophile is green alga Dunaliella salina. That's all from me. Next.
The major difference between eukaryotes and prokaryotes are organisms whose cells are organized into complex structure, and prokaryotes are organisms without a cell nucleus. As I know, eukaryotes have the membrane-bound nucleus, but prokaryotes don't have the membrane-bound nucleus. Furthermore, eukaryotes are multicellular cells, but prokaryotes are unicellular cells. These two are very different because eukaryotes have mitochondria but prokaryotes don't have mitochondria. As I know also, ribosomes at eukaryotes are more larger than prokaryotes. That's all from me.
Let's move to the question number 5, which is about the history of gram staining. So, gram stain is widely used in microbiological staining technique, which is greatly aid in the identification and characterizing the bacteria. So this method, named after the Danish physician named Hans Gram, Hans Christian Gram in 1884, who developed the technique while working with the Karl Friedlander in the morgue of the city of the hospital in Berlin in 1884, and included in his short report the observation that the typhus bacillus did not retain the sting.
That's all from our group. Thank you for enjoying our video.